Good afternoon and welcome. We are delighted you're all joining us. Everyone who's here shares our passion for seeking to overcome problems with hearing loss because we have hearing loss ourselves or a loved one, or just have a passion for really interesting science. So thank you for joining us. We have a wonderful crew of auditory scientists at Stanford, and we're going to share with you some of what's really on the cutting edge of hearing research, and hopefully have some dialogue and interesting discussions with you about what all this means and what the promise of this line of research is. Could I have the next slide, please? So just a few technical reminders. Everyone except for the speakers are muted because there are several hundreds of us. And closed captioning is provided for those of you who have difficulty with hearing. So if you click the button as indicated here, you can turn on the closed captioning. We have received a number of outstanding questions from folks who are online, which we hope to be able to answer today. But if additional questions come up, please post them as indicated. And we may not get to all of them during the live session, but we shall do our best to communicate directly with you and have a conversation with you and hopefully answer your question. And please know that this event is being recorded. I'm Robert Jackler. I am chair of the Department of Otolaryngology, Head and Neck Surgery at Stanford. We have about 55 faculty and several hundred people, and really an incredible group of scientists and clinicians dedicated to overcoming hearing loss. You know, we have created, when I came to Stanford 18 years ago this summer, an initiative to cure hearing loss, which is called the Stanford Initiative to Cure Hearing Loss and is affectionately known as SITCHL. Now, the idea of SITCHL was to bring together in the cauldron of Stanford University the best and brightest scientists from around the world, and then to incorporate with that group of hearing scientists engineers and people from genetics and people from developmental biology to bring to the field of hearing loss some of the very best and brightest scientists and engineers in the world. Now, those of us whose lives have been touched by hearing loss definitely resonate with what Helen Keller said. She had a unique perspective. She was both deaf and blind. And she said, you know, blindness separates us from things, but deafness separates us from people. You know, if you think about it, at the core of humanity, what makes us special as a species of humans is the ability to communicate with each other, to share ideas and our aspirations, to educate our young, to express our feelings. And of course, hearing loss and also voice are core to that. And of course, losing one's hearing is a tremendous loss in the ability to enjoy and appreciate life richly. Now, we know about 2 or 3 of every children born in America are born deaf. About half of seniors have significant hearing impairment. And, you know, today as seniors, and I'm in my mid-60s, you know, I want to live an active life when I'm older as I lose hearing. I want to be able to go to seminars. I want to be able to enjoy restaurants, even when there's a background din. So it's extremely important. And we know that hearing loss has an interface, for example, with decline in mental abilities as one grows older. So the ability to restore hearing, to keep this sense young and vital, is crucial. Now, just a little bit of background, so all of this will kind of make more sense for those of you not quite as up to date on the anatomy and plumbing of the ear. We all see this outer part of the ear, but to an otologist, we look at the sound coming down the ear canal, striking the eardrum, passing through a linkage of 3 little bones to the inner ear, and then the inner ear, called the cochlea, and we're going to pay lots of attention to this structure, sends a signal over the hearing nerve to the brain. Now, when you think about vision, you think about the eye and the retina, which is light sensitive and back. In the inner ear, the analogy is the cochlea, where hearing actually takes place. Now, with apologies to Charles Dickens when he wrote Tale of Two Cities, which I've renamed Tale of Two Ears, in his day in the 19th century, we had no remedy for hearing loss. We couldn't fix anything relating to the middle ear and the hearing bones or the inner ear. Today, however, due to advances in microsurgery in the 20th century, we can fix almost all problems. If you don't have an eardrum or the hearing bones are broken or the bone is stuck, we can fix that. But, as an otologist through my career, I always felt passion for people with cochlear inner ear hearing loss, which is by far the most prevalent kind of hearing loss. And we had precious little that we could do for that. And I wanted to commit my career for making the inner ear problems as treatable as the middle ear and other problems are today. So this is back, before I had gray hair and too many wrinkles, back in 2003 when I came to Stanford. And I decided to partner with many other talented people into a sort of a Manhattan Project type of initiative, which is a large scale, multidisciplinary and goal-oriented effort to bring a biological cure to hearing loss. And that is what we're reporting about today, our progress in that. So let's take a look within this cochlea. Now, hearing research has always been hampered by the fact that the inner ear is so inaccessible and very difficult to study. It's in dense bone. The good news is that we've learned a great deal and have new techniques in the last 2 decades that have greatly accelerated our ability to study the inner ear and to make manipulations that could cure it. Now here we can see a section of the inner ear, and you can see within it these fluid-filled chambers where the sound passes is an area called the organ of Corti. And you see, again, it comes here, and this is elegant drawing shows the hair cells. Now these hair cells, just like rods and cones sense light in the eye, sense vibrations, and that's what sound is. It's vibrations in air, and it's transmitted to the fluid of the inner ear, and this membrane wiggles, causing these cells to bend. And this is what the so-called stereocilia at the apex of the hair cell looks like up close and personal. Now, the inner ear is like a piano keyboard. The high frequencies are at the bottom of the scale, and then as it goes up to the top of the scale, you see the lower frequencies there. Now, this illustrates what's called the traveling wave. Notice that there's a unique point here with this particular frequency is at its maximum. And as that wave moves, it's causing the hair cells to generate a nerve signal. So when this wave forms, it communicates up to the hearing part of the brain, and from there, associations to intelligence and memory and communication. Now, what happens when the inner ear loses hearing? Unlike most organs, it only loses a handful of cells. If you took all the hair cells in your body, they would form a tiny little drop, only about 30,000 cells, 15 per ear. And if you've been in Iraq and had an IUD explode as a young serviceman, it will wipe out hair cells. As we get older, our high frequency areas look like this. But in contrast to every other solid organ, think about the liver in an alcoholic that turns into a scarred lump. But what happens in hearing loss is that almost everything is there. The architecture of the ear remains. The nerve fibers remain. And so really what needs to be done is to repopulate that very limited number of cells that are the things that are core. And this animation shows how many different things, whether it be loud noises or aging effects or toxins or genetic effects, cause the hair cells to go, but the structure of the organ of Corti is largely intact. And importantly, the nerve fibers are there. When people say nerve deafness, that isn't right. In fact, we've long since learned that the nerves are intact and alive. And if you can replace these cells again with new hair cells, the nerve fibers will sprout and link up to them. So there are a variety, as you'll hear from my colleagues, of ways, whether it be genetic or drug therapy, to create new hair cells within the inner ear. Now, I'm just briefly going to mention something you've heard about, no doubt, which is the cochlear implant. The cochlear implant is a very sophisticated device that stimulates the hearing nerve in deafness. What does this mean? A child born deaf today doesn't yet have the ability to biologically repair, which we hope to have in the future. But this can directly stimulate the hearing nerve. So children born deaf today don't go to schools for the deaf. They go to regular schools. They don't have perfect hearing, but this is still quite a remarkable thing that we can replace technically this lost sense. And in fact, this is a product of Stanford engineering and medicine working together in the 1960s and 70s, bringing the first multi-channel electronic stimulator of the inner ear that turned out to be very successful. There are several million implanted around the world. Now, when I wanted to start this Manhattan project-like initiative, I thought, you've got to get the best person. And in 2003, then a professor at Harvard, East Coast School, you might have heard of, and he had discovered stem cells within the inner ear and developed a series of very clever molecular manipulations that could turn them into hair cells. This opened up the field. Stefan was our first recruit, and he and I together recruited many others to build the core of expertise today. So where are we today? In a nutshell, for the mice that are on this computer today, I have good news for you. We're in the midst of taking the discoveries from mice, and a key thing is to bring them to humans. I am a very strong believer that we will make cochlear hearing loss as curable as problems with the middle ear soon. But how soon? You know, I can't tell you whether a child born today would benefit from this as a student, as a high school student, as a young professional, or further in midlife. But I am hopeful that we will make meaningful progress towards clinical implementation in the coming years. Now, one of the reasons this works so well in our group at Stanford is not only because of all the people around us that we can bring expertise from so many different fields to bear with our scientists, but also we're a clinical department. So, you know, basic scientists sometimes want to study the beauty of nature, but we are very focused on understanding those key barriers to implementing human treatment to bring back hearing. And so we have ear surgeons, and we have ear surgeons that work in the lab, and we have purebred basic scientists all working together to common purpose. Now, I have a question for you, and this you'll come up and be able to give your thoughts. Which of the following animals are able to regenerate damaged hair cells? Birds, fish, reptiles, humans, all of the above, or just A, B, and C? Go ahead and make your answers. Hopefully you can see the little screen. We'll give you just a moment. All right. Well, the answer that everyone has said forever has been, yes, birds were the first discovered. That is true. And you know, say, what a miracle it is that, you know, if a bird loses its inner ear, it regenerates. Well, we think of healing. If you cut your skin, it heals. Many organs heal. The human inner ear traditionally has been thought, no, it does not happen. Does not happen. And the correct orthodox answer is all of the above, is A, B, and C. Birds, fish, and reptiles all regenerate the inner ear. Humans don't, except, as you're going to hear at a breakthrough, Alan Chang, a clinician scientist researcher in our labs who's speaking with us today, actually has identified the kernel of regeneration going on in humans, which is a very exciting finding and gives us a glimmer that we can amplify that mechanism. So with that, I'd like to introduce my colleague, Anthony Ritchie. Tony is the chief of all of the basic and laboratory research within our department. He is an associate dean in the School of Medicine over graduate student affairs. And he is a brilliant technical researcher. People come from all over the world to see what he does in electrophysiology. He can record things, single cells. He is a virtuoso of experimentation. So with that, I'm going to go ahead and turn it on over to Tony.
Thanks, Rob, for the introduction. I've never been called a virtuoso before, so that's, I think that's a promotion. So it's a privilege to be here. I'm happy to talk to you, and thanks for showing up and hearing about the work that we're doing today. So in the next few minutes, I'm going to reinforce a bit of what Rob was talking about in giving you an idea of the problem associated with hearing loss. Also, our approach, Sitchel's approach and philosophy, and then introduce you briefly to some of the team members. So as Rob mentioned, hearing loss affects a very large percentage of our population. And the number of people being affected is growing at really an alarming rate. But what's even more frightening is that the onset of hearing loss, when people are really getting symptomatic, is happening to younger and younger people. As you can imagine, this is largely an environmentally driven, culturally driven problem now. But what this means is that more people are living with this problem for longer periods of time, and there remains no cure for hearing loss. You have one set of hair cells, one set of sensory cells, and when they're gone, they're gone. So although hearing loss doesn't kill you, it has major impacts on quality of life and productivity. It also exacerbates other medical issues, and so is really impactful both on the physical and mental health of people. Now, there are many underlying causes for hearing loss, as I'm sure you're all aware, and these are both environmental and genetic, but they all end up really at the same point where there is irreversible loss, typically of the sensory cells. Addressing this type of problem requires a broad range of skills and technologies, as well as scientists who think differently, but are intentionally focused on this common problem. And so we know in this last year, it has been quite difficult, right? With social and political unrest and a pandemic that we're dealing with, and with 2021 starting out kind of as a hold my beer and watch this kind of year. There are a lot of things to worry about, but really there's also been a lot of silver linings. And one of these, I think, is really the clear example of how an intentional approach by a broad team of scientists can really make rapid progress in addressing complex problems. So people from many different fields have contributed and continue to contribute to developing diagnostics and therapies, treatments, computational predictive models, and vaccines, all focused on bringing the pandemic under control in what has really been record-setting time in this development. So the intentional application and focus of scientists with the resources to move forward is really key to progress. Now, this is something that is, I think, hopefully clear to us now, but Rob recognized the power and need of this approach years ago. I guess almost 20 years ago when he started to build this team of researchers that are focused on hearing loss. He also recognized the need to do better. Cochlear implants and hearing aids are still to date the best example of machine-brain interfaces, but there's still no substitute for the biological machine that is the cochlea. As Rob mentioned, he brought Stefan Heller in early on, where Stefan had some very powerful discoveries on the potential for regeneration in stem cells within the inner ear. Stefan came in to help build this team. We're now a team of more than 90 scientists, and it's my pleasure to introduce some of the leaders of this team. So here we have some of our clinician scientists. Alan Chang, who Rob mentioned, is really a cell biologist whose focus is on regeneration. He's also we work together on investigating and preventing aminoglycoside antibiotic ototoxicity. Nick Blevins is also a senior member of the team. He's interested in robotics, and he's particularly also interested in developing diagnostic tools for visualizing inside of the cochlea, and we've worked together on some of that. And Peter Santamaria is one of the newer members of our team. He's a translational scientist with a focus on middle ear infection models. And this is the team of basic scientists. So you've already heard about Stefan and myself. David O'Melody is a physicist and computational biologist. He basically uses mathematical modeling tools to build a framework for interpretation of data that we generate and for helping to design future experiments and identify the areas that we need to move forward in. Teresa Nicholson is also one of the newer members of the team. She's a geneticist and cell biologist, really interested in the pathophysiology at all aspects of the inner ear, and you'll be hearing some of her story in a few minutes. And Nicholas Gurley is a structural biologist and geneticist who focuses on the stereocilia that Rob showed the elegant scanning electron micrograph of earlier. And Nicholas is very focused on understanding the molecules responsible for the mechanotransduction. Now we all come together with different backgrounds and different trainings and different interests, but we're all focused on the inner ear. And we did a little experiment where we had each of us develop a graphical abstract defining kind of who we are and what we do. And this is examples of some of them. And you can see they are aesthetically pleasing, I think, but also you can see how they're all very different, and everyone targets and defines themselves differently because everyone thinks differently. So we're all independent, very successful scientists with a common goal of developing tangible approaches to preventing hearing loss and restoring hearing. And the idea here is that, you know, the whole is better than the sum of the parts, that each of us will be a better scientist for having the others next to us and supporting us. So now it's my pleasure to introduce Teresa Nicholson, a geneticist, as I mentioned, a friend and a colleague.
Thanks, Tony. So, hello, everyone. I'd like to begin by telling you why I joined the Department of Otolaryngology at Stanford. So my lab is interested in the genetics of hair cell function. And here at Stanford, it is a mecca for hair cell research. And this is because of this really unusual combination of expertise. You have electrophysiology in Tony's lab, you have big data analysis in Stefan's lab, you have work on genetics and hair cell regeneration in Nico, Ellen, and Stefan's lab, and you also have computational work being done by DP. Moving to Stanford was really a great way to reinvigorate my own research because of the ability to collaborate with all these different labs. So we study models of human hearing loss using zebrafish as our research model. And this may seem like a surprising choice, but we use zebrafish because they're transparent and you can look directly into their ear, their inner ear, which is something that you can't do with other animals and certainly not with humans. And so what you're looking at is the developing inner ear of a larval zebrafish. And you see these two structures here. These are called ear stones. We also have similar ear stones in our own ears. And this developing ear will morph into this adult structure here. And I think you can see the similarities between this structure and the portion of our ear that's dedicated to balance. So in fish, they actually use this part of the ear for hearing. So if we look deeper inside the inner ear, we can directly view hair cells. And this is in an undissected animal. And these hair cells are very much like the hair cells in our own ears. And here we've engineered a protein to glow brightly in the hair bundles. And we can take this imaging a step further by using software to take a closer look at the shape and the architecture of the hair bundles. And this is almost like an electron micrograph, but it's an image of live hair cells. Now we can do standard methods like electron microscopy. Here, for example, is a micrograph of the face of a 5-day-old larva. Here's the mouth, the two olfactory pits, and then these clusters of sensory hair cells at the surface of the skin. And the fish actually uses these sensory hair cells to sense water movements. And these cells being right at the surface of the skin are fantastic for our experiments because they're so accessible, unlike the hair cells in the inner ear. Another advantage to using zebrafish is the ability to do large-scale screens to look for defects in hearing imbalance. So here I'm just tapping the dish and the fish are darting around trying to escape from the sound. and we look for mutants that don't have this response, such as these deaf mutants here. Now, if you take a closer look at their swimming behavior, they're popping up, they're landing on their heads, they're swimming in circles. Here's even a rare adult mutant also swimming in circles. This is a really strong indication that these animals have lost their sense of balance. And to assess the extent of the hearing loss and the loss of balance in these fish, we can do experiments like play pure tones to the larvae and see how robustly they startle to those sounds, testing their hearing. And we can do things like put the larvae on a moving platform and rotate their heads back and forth and watch the reflexive tail movements in response to stimulation of the inner ear. And here we're using custom software to track the tail movements. So in humans, we actually have very similar reflexes. And it turns out that the majority of the mutations that we have identified are in genes that are associated with human hearing loss. So the fish hair cells require all these different genes for function, and these same genes are required in humans. And in fact, the conservation of gene function from fish to humans is quite remarkable, and because it's so well conserved. For that reason, we can study the defects in fish mutants or in mouse mutants. So both Alan and Nico's lab use the mouse as their animal model. And studying these mutants, we can get a better understanding for the cause of hearing loss. And the reason why this research, this basic science research, is so important is that it provides information about the best therapeutic approaches to use with a patient that has hearing loss. So I should say future approaches because we're not quite there yet. As Rob said, we're on the path towards achieving hair cell regeneration, but there's still some challenges to work out. But I think that gene therapy for hearing loss is something that is right around the corner. And I say this because there's already an FDA-approved gene therapy for the loss of vision. And there's no reason to think that we cannot have the same success with improving hearing loss using gene therapy. So studying what goes wrong in the fish and the mouse mutants gives us a good idea of whether we can actually fix the problem. For example, really severe mutations often leave the hair cells in a state that is beyond repair. So the hair bundles are messed up, the cells are dying. This is a case where regeneration might be a better choice. But we also have mutants where hair cells are more or less normal, but they're not responding to sound. And that's because they're missing a single component, or that the hearing loss itself is progressive in nature. These are situations where gene therapy is likely to work. So one major question is, can we improve hearing loss using gene therapy in mammals? And the answer is yes. So here, the Sichel team is working on optimizing methods for getting the DNA inside hair cells, particularly inner hair cells, which have been resistant to standard methods. I think an important step would be to take these studies into human hair cells, and Alan will touch upon his work with human hair cells in his upcoming talk. Another question is, what is the best way to get the DNA and the reagents inside the inner ear? Because it's a very delicate structure. It is not as accessible as something like the organ of the eye. So, and this is a question that Tony's lab is trying to address. So I'd like to end by saying that knowing what goes wrong in the ear and how to fix it is really a necessary step, a prerequisite that needs to be done before you can use an approach such as gene therapy or regeneration to improve hearing loss. And this kind of work, it really benefits from the input of a team of experts. And we have that team of experts here for SITCHEL, and I'm thrilled to be a part of it. So thanks for listening. And now I'd like to hand it over to my colleague, a very talented physician scientist, Dr. Alan Chang.
Thank you, Teresa. That was a very kind introduction. And hello, everyone. It's a pleasure to be talking to you. Good afternoon, good evening, or maybe even good morning if you're in Hawaii. I've been a faculty and in the Department of Otolaryngology for over 12 years now. I'm a physician scientist, and I practice at the Stanford Children's Health, taking care of children. And as Rob mentioned, hearing loss can be separated into two main categories. What really inspired me to become an ear surgeon is that I learned that the conductive hearing loss, what we see a lot, is something I can learn and fix. But what I became really frustrated was that there were very limited treatment for what we call sensory neural hearing loss. Even though they're very good diagnostic tools, hearing aids, in severe cases, cochlear implants, what I learned is that this is artificial treatment and we do not have a biological cure. So to try to fulfill this unmet need, I made a choice. I decided to become a scientist so I can find ways to regenerate the inner ear. I was very fortunate. I was recruited by Rob, Tony, and Stefan to join the Stanford team. And I chose to come to Stanford for two main reasons. First, the resources of basic and translational research that you've heard about are really second to none in the world. This really allows me to tackle the daunting task of regenerating the mammalian inner ear. And second, as Tony and Teresa mentioned, team science is really how we approach to solve this problem. This is particularly important to me, as I think clinicians working closely with scientists is how we're going to solve this problem. And perhaps most importantly, as you have heard, there's a nucleus of us, both scientists and clinicians, who are passionate about studying how hearing works and ways to reverse hearing loss. So let me just give you a few examples of how we approach this problem to regenerate the mammalian inner ear. As Rob has nicely mentioned, most of the architecture is actually preserved in hearing loss. And within this mammalian organ cochlea, the organ of Corti is where the sensory hair cells are housed. Now, just to put this in perspective, we each have about 15,000 hair cells, and it's the same set of hair cells that have to last our lifetime because there's no natural regeneration. And in most forms of human hearing loss, the fundamental pathology is loss of these hair cells. Just to put this in perspective, there are about 100 billion neurons in our brain. So the silver lining potentially is that 15,000 hair cells to regenerate seems much more achievable. And as you have heard earlier, hair cells are actually highly conserved across species. So for me, I'm really interested in studying mice because this is a mammalian model. And depicted here, as you've seen, as you heard earlier, are the green hair cells. And what in pink here is the cells that are dividing. So you can see that even though there are conservation of hair cells across species, even at rest, there is actually ongoing proliferation or regeneration of hair cells in zebrafish. And these are hair cells actually lying on top of the surface that Teresa has mentioned. Now, after damage, a cochlea in mice is just like human. There is no natural regeneration. But in both chicken and zebrafish, there's regeneration to the point that there's complete recovery of the number of hair cells as well as organ function. So in many ways, these serve as excellent models to study hair cell regeneration. And my colleague Stefan Heller is applying a technique called single cell RNA-seq to look at the mechanisms of hair cell regeneration in chicken. Now, my research program focuses on hair cell regeneration in mammals. We use the mouse model because it offers the tools of genetics, as well as the fact that it closely resembles a human cochlea. Now, shown here, again, green cells are the hair cells that have been damaged, and there is no natural regeneration in the mature mice. But when we look closer, what we found is that the young animals, the young mice, actually have a modest degree of regeneration, as well as proliferation as shown in this picture. Now, to look at this a little bit closer, we actually looked at the other half of the inner ear that houses the balance organs. In one of these balance organs that looks like a pancake, what you can see is that after damage, there is actually some robust degree of regeneration shown in the pink cells here. And this degree of regeneration actually declines with age, just like the cochlea. Now, this is a game changer, because now with these several models, we are now able to study hair cell regeneration in mammals. So to take this one step further, we looked at the balance organ by genetically engineering to express this one protein called ATO1. ATO1 is a gene and protein that is required for hair cell development and is able to give us, has been shown to induce extra hair cells. So shown in green, the little green dots here are the hair cells that after damage, you can see there's a decreased number of hair cells. But by overexpressing this one gene, a single factor, what we see is that the number of hair cells, shown by the green dots here, can be significantly increased. So this can increase the degree of regeneration in the mouse balance organ. And importantly, the degree of regeneration actually correlated to the amount of functional recovery in these experiments. So what this tells us is that regeneration can be a promising approach to restore balance function and possibly hearing loss. Now, so far, what I've been telling you is some of the work on animal models of human hearing loss, in part because the human inner ear is so difficult to access and to study. You cannot get a biopsy of the inner ear without causing any damage. But working with a team of surgeons as well as scientists, we've recently been able to harvest the balance organs of human patients undergoing life-saving tumor removal surgery. This is really a breakthrough. So working with Nicholas Goulet, first we actually were able to use scanning electron micrograph to capture images of individual hair cells. Each of these hair cells is about 1,000th of a millimeter in size. So what I'm showing you here are what appears to be normal appearing hair cells, damaged hair cells, and potentially hair cells that are undergoing repair. So this is really an unexpected finding, that there is any degree of repair or regeneration in the human inner ear. So that leads us to dive deeper into this. Shown here is a cartoon that shows different cell types, hair cells in green and orange, as well as what we presume to be hair cell precursors. These are the cells that can actually regenerate hair cells over time. Now, because there's so few, it's so difficult to get cells from the inner ear, we apply this technique called single-cell RNA-seq that we can look at thousands of cells at the same time. So these are actually individual dots showing individual cells that we can separate into, in cyan here, hair cells, and these are the supporting cells, and right in the middle here are the hair cell precursors that have the ability to regenerate. And furthermore, shown here is a heat map that basically in each cell, we can study the thousands of genes that underlie hair cells, hair cell regeneration. So this really gives us the tools that we can look at the genetic mechanism of how hearing works and potentially how hearing regenerates. So in summary, I've described to you how we at Citro is working as a team to investigate hair cell regeneration. I think we're closer to finding the mechanisms than ever. I think the human models give us a pipeline to really work together to see how hearing works and potentially find new ways to regenerate the inner ear. And on that, I'll pass this back to Tony. Thank you.
Tony, you need to unmute. Thank you.
There we go. Hopefully that's it. Anyway, I'm still Tony Ritchie, now with a voice. My work focuses on investigating the biophysical aspects of hearing at the cell and molecular level. How the molecular machinery basically sets the limit on the function of the whole system. And there are really two broadly defined areas that I work in. One is called mechanotransduction, and this is how sound waves basically are translated into a mechanical force on these stereocilia that Rob described earlier that are on top of the hair cells, and then how the motion of these stereocilia create an electrical stimulus to the sensory cell. And then the other side of this is synaptic physiology. And what that is how does this electrical signal from the hair cell get sent on to the brain? And that's called synaptic transmission that happens at these specialized areas called synapses. And we study how the sound then is converted into a signal understood by the central nervous system. Now, I'm not going to go into details on any of this research. If you're interested in learning more, you can check out my website or send me a note or give me a call. The only thing I'd like doing more than the experiments themselves is talking about the experiments that we do. So feel free. So my interface, what I want to present on is actually a project that I think represents both the power and possibilities of Citro, as well as what I think is going to be a major future direction for research, both within my lab and hopefully within the field. And that is the ability to image live cells in vivo in a functioning cochlea. So to date, cellular-based assays are performed on neonatal cells that are surgically removed from the cochlea. There are very few in vivo examples of how these cells work, and we typically rely on mathematical models to predict what is happening within the cochlea, with no means of actually validating the model because we can't get data from the cochlea. In addition, clinically, only damaged cochleas are really accessed for implants. There are no good drug delivery systems or diagnostic systems. So when I first started thinking about a project to develop in vivo imaging, I talked with a variety of mentors from the past, and they universally said, well, this would just ruin my career because it was impossible. There were too many insurmountable obstacles, and I didn't have the expertise in any of them, and so it wasn't the place for me to move to. So how could I not take that on, knowing that we have the power of the Citro team as well as the university to help support all the things that I don't know how to do. So I think this project incorporates the goals of Citro, and just to illuminate those for you, the goal is to foster high-risk, high-reward projects that are really leapfrogging what NIH is looking for. So they're really too forward-thinking, too risky for what NIH would fund. And I think I've told you how that is. It's meant to seed collaborations between laboratories. So this to basically help build and juice the wheels for lab interactions. So this project has involved working with clinicians and basic scientists, developing surgical approaches, developing a new microscope, as well as developing new genetically encoded sensors and the ability to deliver them. It's meant to support co-mentoring and training of junior scientists, basically to train the next generation of scientists to be better than we are. And I think this project is examples of that because we've had many residents and fellows contributing to developing the surgical approach. We've had postdocs working out the optics and even some engineers working out the optics. And the present postdoc, who's really a rock star now, bringing it to fruition. And finally, Sichel is meant to support infrastructure development for high-end technologies and shared equipment. Kind of, microscopes are to scientists what the water cooler is in many other industries. It's a way to bring people together to talk about what they're doing. And so we had actually an equipment grant from NIH to design and build this microscope. And that's how we were able to get this started. And the only issue with that is that it delayed actually work by about 2 years in terms of just the rate that NIH works. So the fundamental question here, though, is how do you access an image within a pressurized structure that's buried deep within the temporal bone, that is exquisitely mechanically sensitive, yet surrounded by the hardest bone in the body? It's also nearly impossible to deliver drugs or dyes to if you want to have something to image. And it's also very difficult to reproducibly transfect or genetically manipulate in older animals. So let me show you what we're doing. So we kind of turned the problem over and we developed a surgical approach and direction so that we could get at the cochlea with our newly designed microscope. And so we went through several layers of bone to get down to the cochlea. And then we built what we're calling an imaging window. And rather than drilling or scraping or picking at the bone, what we do is we dissolve the bone. So we take out all the mechanical perturbation that would occur by simply dissolving the bone and creating this window, preserving pressure within the ear. You can see here in the cochlea a picture of the imaging window. It's typically 150 to 200 microns in diameter, so it truly is microsurgery. And this is a hearing test called an auditory brainstem response. And all you need to know is down is good. And so, because it means less sound is required to get a response. And the other thing you need to see is that none of these lines are different from each other. That means from before surgery to hours after surgery, we have no effect on hearing. So we can actually make a hole in the cochlea with no effect on hearing. And this is an example of the type of imaging. So this image is from a living, breathing, hearing mouse. And you can see 3 rows of outer hair cells. You can see the tunnel of Corti. You can see inner hair cells. And soon we'll have individual nerve fibers that we can image as well. And so this opens the door now to investigate function in situ of individual hair cells and also groups of hair cells at the same time, something that does not exist in mammal. It is also the ability to deliver drugs now to a functional cochlea. And also, so we've developed that surgical tool. And we've also now worked out ways of genetically modifying cells in older animals. Translationally, this is also really important because it means that potentially we can access the cochlea diagnostically. And so now working, for example, with Nick Blevins on trying to come up with a surgical approach that we can use in the larger human mammal to see if similar approaches can work there. And also now we have a means, we know that we can develop the drug delivery systems that are needed for long-term treatments or potentially for putting in the magic juice for regeneration or what have you. So I think I'm very excited about this. And I also think, future directions will be to do this in awake and behaving animals, which will give us the opportunity to study cognitive function and tinnitus and things like that directly. So now we have a pre-recorded testimonial from one of our Sichel donors and hearing health advocates, Shari Eberts. Thank you.
My name is Shari Eberts, and I am a proud supporter of Stanford's initiative to cure hearing loss. My father had hearing loss, as did his mother. It was genetic and got passed on to me in my mid-20s. I was reluctant to accept it. I had watched hearing loss tear apart my father's life, leading to problems at work, social isolation, and sadness. I was terrified that my life would be the same. So I hid it ignoring my hearing loss and hoping it would go away. It did not. In fact, it got worse and I was forced to get hearing aids. But I hated to wear them. I was scared and ashamed. I hid them as best I could and never mentioned them. But then I had children and everything changed. My hearing loss is genetic, so I worry that I may have passed it on to them. I saw them watching me hide my hearing loss and knew I was passing on a legacy of stigma and shame. This was not something I wanted to do. I needed to accept my hearing loss, so I did. And I have not looked back. I am now active as a hearing health advocate and the founder of livingwithhearingloss.com, an online community for people living with hearing loss and tinnitus. I hope that by sharing my story, I will inspire others to live more comfortably with their own hearing issues. Supporting scientific research about hearing loss is very important to me. Only through research will scientists discover new ways to prevent, treat, and cure hearing loss for the more than 50 million Americans that suffer from it. Most of this research is focused on regenerating the delicate hair cells of the inner ear that are damaged through noise or aging. So when I came across Stanford's work on the genetic predispositions and causes of hearing loss, I was intrigued. I reached out to Stanford and had the chance to meet with the team. I was very impressed. Not only are the researchers smart, creative, and organized, they are also incredibly passionate about their work. I know they will work tirelessly to tackle the very complex research problems involved in the study of hearing loss, and that the team's collaborative approach will accelerate the timetable to solutions. My husband and I have been incredibly pleased to support Stanford's work for several years. We are watching proudly as each discovery takes us closer to the cures and treatment options people with hearing loss crave. I encourage anyone who is touched by hearing loss to consider supporting this important work.
Well, first of all, I would like to thank Sherry for those very kind words that she just shared with us. And I think we have another 10 minutes or so to handle some questions. We got some outstanding questions prior to the beginning in recent days, and we've curated a few of them. And we're going to start with the first one now. But I would also say that we welcome your questions that we'll answer individually and personally offline. So Michelle, could you bring up the first question, please? So the first question is, what causes tinnitus and how is it related to hearing loss?
Well, rock concerts for one.
Yeah, I think we've all had some of that where we leave it and our ears are ringing and hollow for a day or two. It's called a temporary threshold shift. And if you have that a bunch, it tells you you're likely to develop hearing loss. So ringing in the ears and hearing loss are linked. They're like flip sides of the same records for you to be old enough to remember when records had two sides. When you lose the hair cells, the nerve fibers that connect to them begin to rhythmically discharge, even though no sound is coming in. So it is perceived as usually a high-pitched sound, because those nerves are going over and over again, and they are linked to the high-frequency part of the brain. Now, I know we have some really interesting new insights and some promising areas in our science. So I'll turn it over to my colleagues to comment.
So there's been a long controversy over whether tinnitus is central or peripheral. Where does tinnitus come from? And recent data has demonstrated that, in fact, hearing loss can happen without a threshold shift. And so there's often a misdiagnosis when it comes to tinnitus that people have a normal hearing but actually still have tinnitus. Now we know that, in fact, there is loss of input from the cochlea to the brain when you have tinnitus. And further, what's exciting now, and what you have to realize, is that the brain is not a stagnant organ. The brain is something that is constantly responding to the input that it's getting. It's called plasticity, and it's the way the brain adapts to whether it's vision or sound or balance or whatever that's coming in. And so when you change the auditory input, the brain changes its response to that. And at times what happens is it overcompensates. And this overcompensation can often lead to what's called tinnitus. In some ways, it's comparable to tremors associated with Parkinson, where you lose a particular cell type within the brain, and that unmasks a tremor. And what's interesting about that comparison is that there is actually a treatment where they're doing now deep brain stimulation. And what they're doing is they're patterning the stimulus. It used to be that they would just beat the hell out of it. You give you this giant charge and it would take the tremor away for a bit. Now what they're realizing is that they can actually, if they map what the signal used to be and they use a much milder stimulation, they can actually match that and they get much longer term recovery, much longer term masking of the tremor. And so now we have collaborators here at Stanford that are pioneering this, and they're interested. We have a resident right now working with this person, trying to understand what is in fact the stimulus that's lost from the cochlea, and can we bring that in? Can we bring that back in higher up in a similar way as a tool for masking or having the brain readapt itself out of the tinnitus? So what has been virtually an intractable problem, I think, for decades, actually, I think there's a little bit of light at the tunnel. There is some thought now in how we can get at this.
And just one thought is that, of course, if you replenish the hair cells and they take charge of the nerves and bring in meaningful information, so restoring the underlying hearing loss that's linked to the tinnitus. So one question we have is, why does aging cause hearing loss? And why are we, why do some people as they get older lose hearing and other people do not? Alan?
It's a really good question. I think obviously as we age, there's age-related hearing loss is probably caused by a number of factors, including genetics. But I think the lifetime accumulated exposure to sounds is becoming a major concern. Now, we're not talking about necessarily loud rock concerts, but even less noisy sounds that you're exposed to over time, that can be a potential reason.
Well, very good. And I realize that we're now at time, and I just wanted to wrap up. And I want to thank our team that has done such a wonderful job supporting our scientists to be able to come to you today, technically and with clarity. We look forward to further dialogue with those of you who are interested in learning about what we're doing. There's a much younger me pulling on an ear. But thank you for listening. Especially thank all of you for spending an hour with us today. We really value that, and thank you very much. And I want to introduce you to Cliff Harris. Cliff is our wonderful development person. Those of you who support our work accelerate our ability to bring about a cure. There is no question, as we bring in additional scientists and additional advanced equipment, and we draw people from other fields to become interested in hearing, we accelerate the timeline. It's not if, but when. And be able to have that child born today, or those of us who are aging within our lifetimes to benefit from this, is very much helped by resources. And we have a wonderful group of generous supporters who help us. And if you're able to help, we certainly welcome, and you are welcome to get in touch with Cliff. So with that, I want to thank you very much for joining us today. And again, thank our entire team, including our brilliant scientists. Good day.